Yesterday I told you about PAMP and PRR. PAMP is basically a danger signal that the cell recognizes the innate immune system, and primarily the innate immune systems process very fast and tries to keep the infection at bay, honestly speaking, to allow the secondary or adaptive immune response to respond. So there is a time lag that is given by the innate immune system. They basically, the pathogens which are invading, they kill directly and then slow the infection rate and allow the entire immune machinery to be active after the crisis period is over. So it is basically a crisis management, you can say. So this is a rapid action force. There are three types of cells. Remember, you remember this word, antigen presenting cells. The immune system is very much complex because the antigen needs to be presented by somebody else. Suppose you have come to this college, maybe your parents have accompanied or some relatives have accompanied you to this particular place. So at times many students have come here on their own. Many students, most of the students were accompanied by their parents or guardians. So that is the purpose of antigen presenting cell. Antigen cannot be presented directly to the immunocompetent cells. They require to be presented. These are called antigen presenting cells. And there are three types of cells. One is dendritic cell, another is your macrophage. Macrophage is a phagocyte, you know, it kills the bacteria, and the B cells also. Why they are called professional? You are giving an examination, that is called professional examination. University students are also giving examination. Other disciplines are there, but their examination, before that examination, professional award is not there. But in your case, it is there. So there is a definite reason for it. Similarly, their primary job in these antigen presenting cells, their primary job is to present their cells. So you are basically professional because you will be involved in the profession, but there is rigorous training involved in it, which is not present in other systems. So that is the reason why, you try to find out an answer why your examination is called a professional examination, not a simple university examination. Okay. And yesterday I told you about the major histocomparative complex in connection with bone marrow transplantation. Actually, the antigens are presented with the MHC molecules, that is major histocompatibility complex molecules. Now, in connection with bone marrow, it is related to class 2 antigens, but we get infection from virus. So, virally infected cell express antigen, and their antigen is presented by the class 1 molecules. Okay, using the class 1 molecule. Class 2 molecules, they are the actual cells. Okay, so function of class 1 and class 2 molecules is to bind and present to immunocompetent cells. That is the function. Okay, now I am coming to the adaptive immune response after it has come. Invaded that innate system, or it has come directly to the our body. So adaptive immune system will come into play. There are two arms of adaptive. One is your humoral. And one is cell-mediated. Humoral immunity, basically humor means body fluids. So what are present in the body fluids that are related to humoral, and antibody is responsible here, primary machine. In cell-mediated immunity, Basically, it is the lymphocyte. So, T lymphocyte is basically response. This antibody is produced by the B cell, which on activation gets converted into plasma cells. I will tell you in detail about this thing. So, in T cell, there are three types of T cell. One is helper T cell, T helper cell, T cytotoxic cell. This T cytotoxic cell basically kills the bacteria or virus, and there is one called T regulatory cell. This regulate the cell-mediated immunity. Now, those when the actually bacteria or virus, they cannot be managed by the humoral system alone. They are within the cell. Then the cell-mediated immunity is mounted. But exclusively in cell-mediated immunity, not the cells are involved like tuberculosis. It is a primarily cell-mediated immunity. But anti-tubercular antibody is also present in our system. As I told you earlier, both the systems are independent, but they also act in combination. So, this is one example. The TB is basically tuberculosis infection. Basically, we mount a response, and that is essentially cell mediated, but anti-tubercular antibody is also present in the serum. Similarly, we mount immune response here. In humoral immunity, cells are not involved. So, antibody is involved. Why we measure antibody? Okay. Again, another example I am giving, that dengue. Dengue is a viral infection. So, basically it will be killed by the cytotoxic T cell, but antibody will be present. So, dengue, when somebody suffers from fever, and if anything is suspected that he might suffer from fever, so we test the antibody against the dengue virus. Okay, so that is basically humoral immunity. So, the B cells are there. So, B cells actually do not produce antibody. They are converted into plasma cells. So, plasma cell secrete antibody. And there is a process I will tell you in detail. In B cell, while they are producing an antibody, they make a subset of cell, which causes one is antigen antibody producing cell, one is memory cell. But these B cell, while secreting the antibody, also make a copy, and the same antibody is present on the B cell as B cell surface receptor. I will tell you in detail. So, this is basically, now I am explaining it to you, when an antigen, so chronotypic receptor, you remember this B cell receptor. This B cell receptor, what I have said, it is, you will note down, it is a chronotype receptor, is present in single clone of cell. I will explain in detail about these things. So, what happens basically when we encounter an infection, humoral immunity, now I am talking, okay. Now, as I told you that our body has evolved over crores of years of evolution, so we are having all types of receptor in our immune cells. So, when a B cell is present as such, without being exposed to an antigen, it is called a naive B cell. Means no exposure of antigen. Now, when there is an exposure of the antigen, it binds to this structure. Now, that was a single cell. This single cell is endowed with almost 1 lakh receptor. One lymphocyte presents one type of antibody. Okay? So, this antibody, when it comes in contact with this thing, and that is not a simple process, T cells, T helper cells basically present this antigen to this lymphocyte. Anyway, that part I will tell you. So, when it encounters, there is an activation of the B lymphocyte, and from one B lymphocyte, 2 B lymphocytes are produced, from 2 to 4, 4 to 8, just like your PCR. I am drawing a bigger picture. This one you just concentrate. So, up to this stage, 8 cell you have seen. So, from single cell, ultimately we suppose this many cells, again this will divide. So, basically from a single cell, single ancestral cell, there will be an expansion. So, this is a clonal expansion from a single cell. A huge number of cells will be produced on infection, primary infection, when the body is exposed to a infection for the first time. Now, this B lymphocyte which is present there will be converted into plasma cell. Now, this plasma cell will produce antibody. A part of this T cells will be recognized, B cells will be recognized as memory cell. Because now, originally it was 1. Now, after primary infection, the number has increased into n number. N might be 1 lakh, it might be 1 crore, maybe 10 crore. Now, the moment a second exposure occurs, there will be huge response, because this part is now over. The primary response, the antibody level titer will be low, and there will be a lag time for clonal expansion to occur. But when we get a second response, second infection, already a huge number of cells are there. So, they will start producing antibody immediately. Okay. So, therefore, the antibody titer level will be very high in the second response. And actually, during vaccination, you have seen, you have taken a single dose and then a booster dose. Booster dose are adjusted, the time interval are adjusted as per this one, where the clonal expansion is maximum, when the optimal activity will come out. And you remember, during this primary infection, the IgM type, there are immunoglobulin, I will tell you in detail. IgM type of antibody is produced in primary response. From the memory cell in secondary response, IgG type of antibody will be produced. So this is called class switching. Same antibody getting converted. Suppose you got a fever and there is a suspicion of dengue. So the antibody which is tested for dengue is for IgM antibody of dengue. Now, if after investigation, if it is found that you are having an IgG type of antibody, dengue antibody, that means this is not the fever is not due to your dengue infection correctly. You suffered earlier and that's why that IgG level is present. This fever, current fever is for something else. So this is basically clonal expansion from a single clone. Because of expansion, the huge number of cells have been produced. That is called clonal expansion and memory creation of memory cells. So since this produces antibody and it is of homogeneous in nature, while producing this thing, one copy, one molecule of the same antibody remains membrane bound, and that is called clonotypic receptor. Clone, single clone of cell which is producing this thing, that's why the receptor which is present, it is called a B cell receptor. B cell receptor is essentially clonotypic receptor, and this is an immunoglobulin which the same B cell has produced. Now you see the B cell is basically lymphocyte. Now on activation, clonal expansion, basically a plasma cell is produced and one subset of cell remains as a memory cell. This memory cell responds very fast. Okay. So you see, this is the clonal expansion I am telling you, these are the effector cells. This is PC, means plasma cell. This is producing antibody, and you see there is a memory cell. Similarly, in the T cell also, same thing happens. Only thing is that a subset of cell remains as a memory cell in the form of T helper, T cytotoxic and T suppressor. The main function, so the main function of T helper cell is to help the immune system. T cytotoxic basically kills this thing, and T regulatory cell basically regulates the immune response. Now you see this response, this one. So primary response, so there will be a lag period, because lag period is here, because this phase, from this phase to this phase, we require some amount of time. It might be 5 to 7 days, it might be 30 days also. You have taken booster dose at monthly interval. Remember, all the vaccinations are given usually at a month interval, a bit later. So the time course, this is the time course. Now when the primary response has occurred, now a second immune exposure is there, and you see this is the antibody titer. Antibody titer has increased like this. Okay. So this is the actual curve for primary and secondary response, and there is a term called negative phase. Now during this secondary infection in the early part, if a second infection occurs, because the antibody will be neutralized, there will be sudden drop of antibody, called a negative phase. So negative phase is present in the adaptive response. Since there is no antibody, it is not present in this thing, in the innate immunity system, and it is not present in the primary system as well. Okay. So coming a little bit back, this I have told you, this so adaptive immunity takes about 4 to 5 days minimum time, because to develop, and it is a highly specific for the infectious agent, because a specific immune response has been mounted against it and display the specific type of receptor on the plasma membrane. And when the second time the infection occurs, there will be a rapid response. Okay. So in innate system was immediate, adaptive system was delayed, and this delay is because of this procedure. Okay. So why it is called B cell? Because originally the T lymphocyte has been divided into two parts. One is B lymphocyte, one is T lymphocyte. Because it was discovered in the cloaca and the bursa of Fabricius. That is the reason why it is called a B cell. But in our human system, it is derived from the bone marrow. So bone marrow we get the B cells where they mature. They are named B cells. Named B cells means they are not exposed to any antigen. Then they are processed, and then they become immunocompetent cell, and then they produce the antibody. So the B cells per se do not produce antibody. B cells gets converted into plasma cell produces antibody. Now when there is a cancer of the plasma cell, abnormal type of immunoglobulins or immunoglobulins will be present in the high amount, and that cancer of the plasma cell is called multiple myeloma. And if it is localized, then it is called a plasmacytoma. It can be a bone, it can be in a soft tissue. So basically, the plasma cell, where it is, when it becomes cancerous, the cancer cell may produce one type of antibody, or it might contain various types of antibodies, and that will produce a high amount of antibody because this is not a normal cell. So their numbers are high, so they produce an M band about which you have read, and I will tell you at the if the time permits. Okay. So basically, you remember, if you have read this thing, hematopoietic cell is divided into two groups. One is lymphoid series, one is myeloid series. The cells which are from the lymphoid series are involved in the humoral immunity. The cells which are produced from the myeloid series, that basically neutrophil, eosinophil, basophil, natural killer cell, dendritic cell, antigen producing cell, they are involved in the innate cell, innate immunity. Now a little bit of function, only one function you write down, only this part you write down, okay. There are several, only just note down this one. So phagocytosis is NATE means neutrophil extracellular trap. What is neutrophil extracellular trap? I am just telling you. So neutrophil basically this is an extracellular DNA molecule along with antimicrobial substances which traps a pathogen. That is called a neutrophil extracellular trap. Okay. So they won't, try to manage the pathogen or kill the pathogen through one mechanism, that is called neutrophil extracellular trap. Now coming back to this thing, these are the immunocompetent cells of the innate system. We will note down only this thing, phagocytosis and NATE. It is done by the macrophage and neutrophil. One line at least you will say in the innate immunity. Histamine production is by the mast cell and eosinophil. Cytokine production, this mast cell, dendritic cell, lymphocyte, eosinophil and basophil, and parasitic attack, you can see the eosinophil and basophil. They attack the parasite cell. These 4 points at least you note down. Nothing more than that is required. Rest you can read the book. I want that you write something about the innate cell, what is written in your book, at least the function of these cells, so that it becomes a complete answer to some extent. Okay. So B cell produces this receptor, as I told you, that is called a clonotypic receptor. Similarly, T cell is also having a receptor that is called a T cell receptor. They are highly specific to recognize antigen. Okay. So this part I told you, the B cell has almost 1 lakh antibody molecule on its surface. In contrast, T cells usually have 30 to 40,000 of receptors on its surface. So can you remember, one cell is having so many receptors? The cellular structure, what we have said or read, is not that simple. It was not that schematic. It is much more complex than what we really understand. The more you read, the more you realize the complexity. So, humoral immunity and cell-mediated immunity, they are basically controlled by T helper cell. T helper cell basically present the cell, present the antigen at one side to the B lymphocyte, the other side to the T cytotoxic cell. And that is the reason how both the systems, T helper cell is basically acting as a intermediate cell between the two systems. So, when any antigen or bacteria comes in contact with the body tries to engulf it and then tries to destroy. After destruction, some part of the antigen is expressed on its surface, and then basically the T cell come into contact with it, then the T cell get activated, and that activated T cell binds with the B cell, and then the B cell actually gets this clonal expansion occurs because of this thing. And after the clonal expansion has taken place, a part of this thing will remain as a memory cell, which is designated as this color. The rest of the things, these are basically produces antibody. Okay. So this is basically class switching. So just remember, IgM indicates a recent infection. IgM of any bacteria, virus or anything, detection by ELISA method or any other method indicates a recent infection, but IgG is a old infection or previous infection. Okay, this part I have told you, this part now you see, in your chart you try to understand the meaning. So first, immune response, primary. Now there are two in the adaptive system. There are two things. One is primary, one is secondary. One is secondary. Okay. So primary, the immune response against first antigen. First one it is getting exposed. The second against the subsequent exposure. That is secondary response. Antigen processing, both T dependent by the T helper cell, as I told you. It can be T independent also, as I told you also, that some of you have come alone, some of you are accompanied by your parents or guardians, something like that. So all these things does not require. So only this secondary infection is T dependent. So lag period, there is a lag period, and that is variable in primary response. It can be 4 to 5 days, it can be 15 to 30 days also. Now, there is no lag period here because already cells are ready. Huge number of cells are ready and they will go on producing antibody. So, there is no lag period. Antibody type, again I am repeatedly telling, the primary response it is IgM type and secondary it is IgG, and there is a class switching during the secondary response from IgM to IgG because of the genetic rearrangement. Antibody titer, as you have seen, the antibody titer was low in the primary and it was high in secondary. Avidity is another issue. How avidly the antibody binds to the antigen. It is less in primary, but it is more in secondary. And nature of response, triple S, it is slow, sluggish and short-lived because of this thing, this structure which I have shown you. And it is a prompt secondary response, and it is powerful and prolonged. So the 3 S is there, 3 P's are there, 3 S's are there, 3 P's are there. You remember in that way. So passive, now you have, by this time you have known about the active immunization, what is basically an adaptive immunization. You have acquired it. So it will be definitely a long-lasting one. In contrast, if something is given to you as antibody directly, so that will have a short life basically. So here in the passive immunization, there is no booster dose required. But for active immunization, booster dose is required. Lag period, there is no lag period because you are giving directly the antibody. So it is present here, duration is short lasting. As you can see, the life of that antibody will basically determine the duration of immunity. Memory cell, of course, it cannot be because there is no system, but here it is memory cell is yes. Usefulness in immunodeficiency state. See, all persons will not remain normal, they will be diseased. they will fail to mount an immune response. So, when they suffer from infection, immunoglobulin is to be given to them. So, immunocompromised cell or immunocompromised patient or immunodeficient patients are given antibody as a routine to protect them, otherwise they will suffer from infection. So, innate adaptive, you by this time know in detail all the points, whatever I have told you, I have explained it in detail in the last class and this class. Now, this chart will be very much easy to remember all the points. Cell-mediated immunity, I told you the 3 cells are involved, basically T cytotoxic cell. You see the T helper cell, T cytotoxic cell and regulatory T cells. These are the 3 types of cells, and of course, just like a B cell, humoral immunity, there will be a memory T cell because it has to handle with the future infection. So, basically it is involved in the direct killing of the bacteria or viruses or infected cell or the cancer cell. It plays a major role in destroying cancer cell. Remember, the function, this is basically there is an activation of phagocytes and then production of antigen specific cytotoxic T cell. Because cytotoxic T cell is the T cell which kills the bacteria or virus, and then the release of various types of cytokines, which is beyond the scope of this short time of this class. I cannot discuss it, but cytokines basically are interleukins. These are basic, one full class will be required on cytokines. So, cell-mediated immunity, major functions of T lymphocyte. Just note down the point if you can remember, otherwise you read it. One is immunity against infection, immunity will be given, definitely allograft rejection. T cell plays a very important role, and the tumor cell destruction. B cell cannot kill the tumor cell. Cytotoxic helper function. T helper cell basically acts as an intermediate cell between the humoral immunity and the cell. And the suppressor function is to regulate the T cell, because excessive immunity we do not want. We want the desired level of immunity. So, that is why there are kind of some suppressor function is there. Helper T cell helps in boosting immunity, suppressor cell controls or regulates the level of immunity in a given individual. So, basically you can see the T helper cell, this is the T helper cell. This T helper cell is presenting the antibody to the B cell. Now, macrophage here is, macrophage has engulfed the bacteria, it has killed, and a part of the antigen has been expressed to it, and it is giving to the T helper cell. So, that is the basically central role. Now, functions of the T helper cell basically are the number of this thing. Now, I cannot spare more time on this thing. I have to remember tuberculosis is a cell-mediated immunity, and in clinical practice, you will do advise somebody suffering from tuberculosis, go for a tuberculin test or called a Mantoux test. So, that Mantoux test is done using the tuberculin antigen. It is given subcutaneously, and then the response is seen. This is a classical example of cell-mediated immunity. This is, it remains intracellularly. Cell cannot destroy the tuberculous bacillus. That's why cell-mediated immunity is mounted. But if you try to measure antibody level of TB, yes, it will be present and it can be detected. Missing cells. So, the antibody, now coming to the antibody, which is the main part. Antibody is basically a biomolecule which is produced in response to antigen and is capable of binding to the antigen. Its main function is to neutralize it. Okay. So, I told you yesterday there are specific regions, all the regions of antigen do not interact with the antibody. Epitope and paratope I have told you. So, this is the antigen. Suppose this is the antigen. This is called a epitope which reacts with the antibody. This antibody binding site, I will tell you in detail about antibody binding site. So, this particular region is called a paratope. Okay, so epitope and paratope determines the specificity of the reaction. If there is no interaction, it cannot be. I'll spend a little time about this thing. It is very easy to understand. Only thing is that you remember the points. Basic structure of an immunoglobulin. It is a Y-shaped structure basically. Okay. This is a Y-shaped structure. It is composed of two heavy chains. The structure which I have drawn is basically a heavy chain. There are 5 types of heavy chain. One is alpha. So this is an immunoglobulin. So alpha means A. Gamma, alpha beta, gamma means IgG. Mu means IgM. Delta means IgG, IgD, and epsilon means IgE. Okay. So these chains are named as per the gene. So if it is an alpha, then the 5 types of immunoglobulin is there: IgA, IgG, then A, then M, D, and E. Okay, these are the 5 types, and in each immunoglobulin molecule, 2 light chains are present. In this part, if I put my hand like this, both the hands, these 2 are heavy chains, and this is a hinge region. This moves, and this is the region which binds the antibody. So this part is divided into 3 domains. One is here, that is called CH1, that is C means constant region. This is the constant region, and this is called the variable region. This is called the variable region. Okay. So this is constant region 1, this is constant region 2, this is, or you can write down in this fashion. Here I am telling you this is light chain, okay, and this is heavy chain. This light chain can be of kappa or it can be of lambda. But in a chain, in a immunoglobulin molecule, either kappa will be present or either lambda will be present. So either 2 light chain will be lambda or 2 light chain will be kappa. That is fixed. So mostly kappa chain is present almost in 60% cases, and rest 40% cases lambda is present. Now there are 3 domains in this heavy chain, that is called, C means constant region, heavy chain is there, so CH1, CH2, and this part is CH3, and this is variable region. Okay, the variable region I will tell you in detail. Now there are disulfide bonds here. There are disulfide bonds here, disulfide bonds here. This part, so I am just representing it with this small structure. Okay. So this part is called fragment antigen binding. Because this part, this region, this variable region basically binds the antigen. So this entire part is called FAB region, fragment antigen binding. Since there are two antibody binding sites, so it is called FAB2. And this portion is called FC, means fraction crystallizable. Crystallizable, why it is called? Because many proteins are structure is done by the crystal X-ray crystallography. So that protein needs to be crystallized. So this part is crystallizable. Now there are two disulfide bonds here. Important part is here, cleavage by pepsin and cleavage by papain. Pepsin, if you remember correctly, it hydrolyzes the carboxyl terminal end of basically aromatic amino acid and some hydrophobic amino acid. So, pepsin, remember there are two things, pepsin, pep1, sin1. So, pepsin bonds basically cleaves here. So, you get one FC portion, one part which is having two FAB fragments. So, basically pepsin, when it is cleaved, a little just below the disulfide bond, it gives two fragments. And if you cleave it with papain, PAPA, remember this PAPA and in. So there is one fragment here, one fragment here, and the one fragment will be here. So it will cut the structure here, near here. Once the immunoglobulin is cut, then one will be FC portion and one will be FAB portion. Two FAB portions will be there. So papain will cut and make three fragments. Now, why it is important? You can ask, what is the purpose of knowing this thing? It is not written in your book, I am telling you. We want to see the function. It is for research purposes. We want to see what is the function, which part of the antibody molecule binds what type of function. Whether it is in fragment antigen binding, now this fragment will bind with the antigen, not this one. If you incubate, then only we understand. And how many antigens are being bound? Now, if you cut it here, if we fragment with the antigen, now these will not bind with the antigen, but these will bind with two antigens. Now, if I cut it here and then make three test tubes and incubate it, we will find that this one is binding one, this one is binding one, and this one is not binding. Now then, what is the function of this one, FC portion? It basically is a glycoprotein. It acts as a receptor and it binds with the actually macrophages or other, there is a part called antibody dependent cell-mediated cytotoxicity, ADCC. So this part is to be recognized by the cell surface. And there is one important part, the complement activation. Complement activation is a part, complement gets attached to this part. This part. Now again, if we want to see the complement activation study, this complement activity study will not be possible with this one. So we need to fragment this thing. This will be basically for the pure research purposes. So now coming to the basically different, there are 5 different types of antigen, antibody, that is IgG, IgM, IgD and IgE. This is the basic structure, you remember. One is Y. This is the structure for the IgG. This structure is called IgG, and its molecular weight is about 1,50,000. Okay, so it cannot cross the, globular filter. It cannot. It is impossible. Actually, this is the antibody which crosses the placenta and gives immunity to the baby, and this is the antibody which is present in the secondary immune response, not in the primary. Now, when 2 units of IgG or 2 units of such type of structure are added together with a joining chain, then it becomes IgA. So, IgG is basically a monomer, IgG is basically a dimer, and it is connected by a joining chain. And this IgA is present in the mucosal secretion. It gives surface immunity in all secretions we are having, including breast milk. So, we have IgG. This IgA, when the baby takes the milk from the mother's breast milk, basically, it gives an intestinal immunity, local immunity, because it is present in the as a surface immunity, saliva, tear, gastrointestinal secretion, other body fluid secretion, IgA is present, and it confers mucosal immunity. And IgD function is not that much known. It binds with the immune system and it confers immunity also. IgE is related to allergy. Ig level is the minimum level present in the blood. Now, those who are suffering from allergy, first investigation is done, measure the level of IgE. A high level of IgE indicates the patient is, what the complaint is there, it is basically due to allergy. Now, you have to give the treatment and you have to follow up whether this level has decreased or not. Otherwise, so this IgE is basically released from the mast cell and it is concerned with the allergy reaction. IgM in contrast is a pentamer. So, you see here there is a 2 binding site. So, IgG, IgD, IgE will also have a 2 binding site. IgA will have a 4 binding site. And now IgM is a pentamer. So, 5 such structure will be there. So, they will be joined together. First and foremost, they are joined to each other by disulfide bond. In addition, there will be J chain. So, now you see the IgM is a pentamer. So, it is having 10 binding sites. And this IgM is produced during the primary response. IgG is present in the secondary response. Ig is a very powerful agglutinating agent. See, when antigen and antibody reacts, they make a precipitate. They are thrown out of the solution that causes precipitation. Precipitation is made by this IgG because of the antigen antibody reaction. Now, I think you have taken an antibody blood group testing, you have seen it curdles. Basically, this is an agglutination reaction. When a particular matter reacts with the antibody, it gives a clumping, visible clumping, and that is called agglutination, and it is done by the IgM type of antibody because 10 antigen molecules are getting attached to each other. So, this is called an agglutination when it is combined with the antibody that causes agglutination. Agglutination is a large structure. Now you understand, if we coat some particulate matter with this IgM type of antibody and we do an immune reaction in vitro, in the test tube or on the slide, we will see a visible clumping, but that visible clumping will not be present in the precipitate. We can allow the reaction to occur, then we need to centrifuge it to see the deposit, whether the precipitation has taken place or not. But here you will see a visible clumping right on your slide itself. Okay, normally it is done with the latex particle for some purposes. So for some testing, you can this and latex particle are coated with the IgM type of antibody to see a visible clumping right on as a spot test. You need not require to centrifuge it. So that is the difference between the precipitation and agglutination. So agglutination occurs with the IgM type of antibody, but not in other type of antibodies. So it is of 1,50,000. Now if two molecules are attached to it, so it will be 3 lakhs almost. Now IgM, there are 5, so 1.5 lakh into 5, it will be minimum 9 lakhs. In addition to there will be J chain also. So some weight will be there. So normally to remember very easily, we remember that this IgE is 1 lakh, IgM is 9 lakhs, and IgD is 180 lakhs, and IgE is 190 lakhs, because there will be 1,90,000 and 1,80,000. Because there will be structural difference. So similarly, amino acid sequence will be different, and all the structures will not be having the same amino acid sequence because of the antibody structure of all against all antigen will not be the same. So there will be some, range of variation. So this part I think you are clear. So this is basically the variable region. This basically is the antigen binding site, not the heavy part. Although this is called an FAB fragment, but this part is concerned with the antigen binding, and this is much more complex than I will tell you shortly in detail. So this variable region of IgG, the first 100 to 110 amino acids are involved. This is the N terminal. This is the N terminal of the polypeptide chain, and this is the carboxy terminal. Just remember C. This C means this is carboxy terminal, this is amino terminal. You draw a picture of immunoglobulin and try to understand, it will be easier for you to understand. Okay. So as I told you, second, these domains I have explained all these things. This heavy chain, light chain, the functions of IgG, you at least remember the first part is agglutination or neutralization of the toxin. Second is opsonization. Bacteria is having very bad surfaces. So phagocytes, if they want to kill, their plasma membrane will be destroyed. So they coat that foreign material or bacteria virus with the IgG type of antigen, that is called an opsonization. Coating the surface of the pathogen is called opsonization. And there is activation of the classical pathway, neutralization of toxin, of course. This is most important thing, and that is the reason why the antitoxin or anti-IgG or in the previous years anti-tetanus serum, what we used to give basically to neutralize the toxin produced by the bacteria. And antibody dependent cell-mediated cytotoxicity is an important topic. I could not cover this thing. So this structure I have already told you. IgG structure is a monomeric structure, IgG, IgA, D and IgE is a monomeric structure, IgA is a dimeric structure. It can occasionally exist as a monomer, but it is a dimeric structure joined by J chain. IgM is a pentameric structure having 10 binding sites joined by the J chain. So now you see the level. Normally, since we mount an immune response to a whole large variety of antigens, so IgG level becomes very high. This is of the order of 1000 milligram roughly, but it varies. Now we are not allergic to many things, and that's why you look at the level of IgE, it is level is only 0.05. Now, when somebody is having an allergy, first investigation you do an IgG testing, and the level will be definitely high, very high. Depending on the type, depending on the degree of allergy he is having, the level will be high, and the patient will be too much conscious about cell level. আমার IgG লেভেলটা বেড়ে গেছে, আমার সেই জন্য অ্যালার্জি হচ্ছে, ঠিক উল্টোটা। তোমার অ্যালার্জি হয়েছে তার জন্য IgG লেভেলটা বেড়েছে। আমার খুব সর্দি কাশি হয়, আমার ইউসিনোফিলটা বেড়ে গেছে। সর্দি কাশি যেটা বলে নর্মালি, ইট ইজ মোস্টলি নট ডিউ টু কোল্ড, ইট ইজ ডিউ টু অ্যালার্জি। Because of the allergy, eosinophil count has increased, not the other way around. People say, আমার eosinophil আমার বেশি আছে, সেই জন্য আমার হাঁচি কাশি হচ্ছে। It is the effect of that thing. So don't try to get confused. I mean, with the patient statement, you try to explain, okay, fine, agreed. It will be difficult for anyone to explain that it is due to your disease process. It has increased. This is a marker. So this part I have told you, the agglutination will be very strong with the IgM type of antibody, but IgL will be intermediate because there are 4 binding sites. But it will be very strong here. The primary response, the IgM, remember always, I have told at least 10 times, and IgG is for the secondary response. Okay, placental transfer is a very important thing. Mother confers immunity through IgG, and complement fixation is very high with IgM. IgG also causes complement fixation. So this chart is available in your book. You just read it. Only thing is that I have highlighted the points only to highlight the areas. This you read it. There is no. I have already explained. Only thing is that you have to memorize all the points. Now, why we are having this part, I will take another 10 minutes time just to explain this thing. See, why innumerable antibodies are produced. What is the basic mechanism? It is basically because of you just write down one thing. These 2 lines you write down about the antibody diversity. This part, and the second slide only. These 2 to 4 lines will be there. So how innumerable immunoglobulins are formed? We write down there are about 150 immunoglobulin genes, and these are basically variable region genes, and there are almost 1 million antibodies are produced. It's basically by a variety of joining of the VDJ region. This region, what we have said as V region, one is D, one is V, one is J. The diversity is basically due to this gene. D is standing for diversity. Now, by a variety, there are 3 sets of genes are there. B, D, and J. Now, there will be a combination of all these things. Because a high number of genes are there, so they will make a combination and they will produce a variety of antigens. Write down by a variety of combination of variable region, diversity region, and the joining region, leading to VDJ joining of the H chain and the light chain. Okay, this is the basis of antibody diversity. You just focus it here. I am telling you. So, I tried to look what is the number actual number. So Wikipedia said about 30,000 crore. Harrison latest medicine, I have also checked it. It is saying that Vasudevan book, it is 100 crore. So anyway, 100 crore is not a matter of joke. Second line you write down. This only one line down. The somatic recombination of multiple germline gene segment, not the somatic gene segment, due to exposure of antigen during individual's lifetime. This is the basis of antibody diversity. These 2 lines will be sufficient to explain and try to understand. You see, this just try note down, and I'll go to the next one. It will be clear to you. So now you see there is a VDJ joining region. So there are 4 genes for explanatory purposes I have written. There are 4 genes here. Now if this portion is deleted, we will get a combination like BDJ joining like this. Now if we delete this part, we will get a combination of this variable region is just like that. So it depends how the cell will process. This is a splicing reaction basically. Post-translational modification somewhat. And the genes are located, you see, the genes are located at different places. So I have checked from many sites what are the different, how many actually genes are present, and multiple sources, I get confused. And Sushma Tonegro was the person who got Nobel Prize in 1987. And fortunately, I read his entire research paper because in my third paper, MD examination, this question was given and there are 16 questions and 16 questions have to be answered and all 16 questions should be checked by 4 different examiners. And if any question is not addressed properly, substantially, then they fail. And you can look down, look at it, there is no marks for the MD examination. It is purely an assessment, a classical example of assessment, whether you are pass or whether you are fail. So if I could not have written this properly, obviously I would have failed. So this question is there. So antigen antibody binding is basically the same thing. It is not a covalent binding, same electrostatic band, van der Waals force and hydrogen bond. Disorders of immunoglobulin I cannot actually cover because if your time is over, you have to go for the other classes. So can you spare 5 more minutes so that, can you spare? So I just complete it and you read it from the book. I am just telling you salient points only, which may not be present in your book. So M band, you know, during plasma protein, I think it has been told, M band, was it covered, M band? Are you aware of it? M band means multiple myeloma band, okay? Multiple myeloma, M band. During plasma protein class, I think you have been told. I am just telling you. So this is lymphocyte, they get converted with plasma cell. When the plasma cell becomes malignant, it becomes basically a multiple myeloma. And when the multiple myeloma occurs, the gamma globulin portion will be increased. So on electrophoresis, there will be a band, and this might come as a spotter also. You just try to understand this one. This will be given. One set is there, and one spotter is expected. So that is the reason I have told you. So if you do an electrophoresis, you will get a band like this. You read it. If you cannot understand, you can come at any point of time to the department. I will be happy to explain it to you. So this is the basically, if you do an electrophoresis, albumin is the smallest molecule. So it moves to the anode. This is the albumin molecule. Now this is the gamma region. So gamma region may be this is absent. That means there is no gamma globulin, a gamma globulin. And this is a normal thing. Now you see there is a band here. This is indicating that levels of gamma globulin is increased. Now you see there is one part here at the beta position that is called a beta myeloma. It is a myeloma, but because that cell, cancer cell, has not produced the desired type of antibody, because this is a cancerous cell. So it has produced an antibody which is of that size, which is moving with the beta globulins. That's why it is called a beta myeloma. It can be a beta myeloma, it can be a gamma myeloma. Okay. Now multiple myeloma, M band is related to basically, multiple myeloma. It is basically due to cancer of the plasma cell. Now when it is localized, either the soft tissue or in the bone, that is called a plasmacytoma. In both the conditions, a immunoglobulin level will be very much high. So you see this picture will come, there will be a spotter for this. What is the diagnosis? This is the band, you see that the beta position, there is a band. So this is a beta myeloma. Okay. So you see this is a band, this is a beta myeloma. You see the peak here, M band. So M band is corresponding here. So a myeloma protein, write down a myeloma protein results from monoclonal proliferation, because that is again a single clone of cell. That's why there will be monoclonal proliferation of cell, a single cell, and that will produce a homogeneous type of antibody. Now, if by chance, the cancer cell produces smaller fragment of light chain, the light chain being smaller in molecular weight, it will pass through the urine. It will be filtered by the glomerulus. So that will cause another type of protein that is called a Benz-Jones protein. So Benz-Jones proteins are found also in multiple myeloma, but in my 31-year career at Durgapur, I have seen several cases of multiple myeloma, but I could not detect it. Any case of Benz-Jones protein, then I got a clarification in the conference that it is very difficult to get it. You just go for immunofixation electrophoresis on a concentrated urine. So when in future you are planning for evaluation of a multiple myeloma case, do not ask for Benz-Jones protein, because this technology was not present. Immunofixation electrophoresis has come from the 90s, because during my MD study period I know it. So immunofixation electrophoresis of the concentrated urine is a desired investigation. Most of the doctors even do not know this thing. Okay, this is a superior technique. So disorders of the function, it can be either hyperfunction or hypofunction. So if there is a hypofunction, that means if the immunoglobulin is not, cell is not produced, or the T cells are not functioning, then the patient will be suffering from infection. And hyperfunction, yes, it can occur if there is excessive immune reaction. It can occur in multiple myeloma. It can occur in chronic active hepatitis, and there will be AG ratio reversal. Remember, AG ratio reversal occurs in 3 conditions. One is multiple myeloma. The uncommon, common is basically nephrotic syndrome. Nephrotic syndrome, albumin level will be low, globulin level, that's why globulin level will be high, and that is the reason for nephrotic syndrome. For multiple myeloma, it is because of the increase of the gamma globulin. Albumin synthesis will be compromised if the liver is affected in the cancerous process. And another thing is that chronic active hepatitis, because of the hyperactivation of the hyperfunctioning of the immune system. Okay, so T cell function, if it is destroyed, then T cell will not operate. B cell function, if it is destroyed, then B cell cannot occur. So how will you diagnose it? There is 3 diseases only I remember. One is common variable immune deficiency. You do a electrophoresis and you will study whether what type of immunoglobulin is present or not, whether it is an IgM or IgG type of antibody. So primary, secondary, severe combined immunodeficiency is another one, but T cell, B cell both are damaged, and patient usually cannot survive. And this is called severe combined. This is called bubble baby boy, because these boys, if born, will not survive. They are kept in an incubator in a plastic bubble. That's why they are called bubble baby. Because once they are exposed to the environment, they will suffer from infection and they will die. So that is the reason why it is called a bubble baby syndrome. There is a movie for it. So usually death occurs, very rapidly within a year. So primary antibody deficiency will be there, but these are very, very rare conditions, I am telling you. So investigation you will do IgM loss or IgG loss,